Good evening. I'm Daniel Benjamin, and I'm the president of the American Academy in Berlin, and I'm delighted to welcome you to this spring's Kurt Viermet's lecture entitled Some Assembly Required, Decoding 4 Billion Years of Life from Ancient Fossils to DNA. And our speaker tonight will be Neil Shubin, and that is the title of his latest book. It was published last March in English, but we are doing this tonight to coincide with the publication of the book in German. It's being published by S. Fischer Verlag, who is our cooperation partner for tonight's event. And I want to thank the Verlag for its cooperation, and hope we can do this many more times in the future. The Kurt Viermet's Distinguished Visitorship, which Neil Shubin holds, was established in honor of the late Kurt Viermet, who was the retired vice chairman of JP Morgan, in recognition of his exceptional career in international finance and his dedication to the transatlantic relationship. And the fellowship was inaugurated in 2008, and it has brought an array of distinguished scholars to the Academy, and we hope that at some point Professor Shubin will be able to join us live, but we're delighted that we have the technology so that he can join us virtually. So my job is not to introduce him. I'm going to hand that over to Professor Madeleine Beekman, who will be the moderator for tonight's lecture. I just wanted to say how excited I am about this talk, because I realized in reading up on Professor Shubin that he was a student of one of my all-time favorite teachers, the great paleontologist and evolutionary biologist Stephen Jay Gould, who unfortunately died young, but was a hero to, shall we say, the popular science world as well as to the serious science world in the late 20th century and the very beginning of the 21st. So I'm thrilled about tonight's event. So Professor Madeleine Beekman is professor of ecology at the University of Sydney. She is currently a fellow at the Wissenschaftskolleg to Berlin with a project entitled How Evolution Works, From Genes to Organisms and Back Again. And in that project, she explores the concept of survival of the fittest and contrasts evolution at the molecular level with evolution at the organismic level. Professor Beekman is part of the so-called B Lab. That's an acronym for Behavior, Ecology, and Evolution Lab, which initially focused appropriately on researching bees, but now includes other organisms as well. They study those organisms to see how societies suppress selfish behavior by individuals, as well as evolutionary genetics and host parasite dynamics. So Professor Beekman, we're thrilled that you could do this event, and we're thrilled to have you here. And the virtual podium is now yours.
Thank you very much. And also thank you very much for the introduction. And it's nice to hear about my own project here, because it has changed a little bit. So this is sort of a ghost from the past. Well, it's my great pleasure to do the introduction today, because a few years ago, probably actually quite a few years ago, my husband gave me a book, and he told me, you really have to read this book because it's amazing and it's about evolution. And of course, I read that book and I completely agreed with him that this is an amazing book about evolution. And of course, that book was Your Inner Fish. So you can probably imagine how I felt when the Academy contacted me to ask me if I was happy to introduce tonight's talk. Because it's a great delight to be able to do this. And it also pushed me to read Some Assembly Required, because I hadn't kept up with all the wonderful books that Neil has been writing. And it's so nice to be here for the German translation, although I do prefer reading it in English, which I have done. Neil was trained at Columbia University and Harvard, especially at Harvard. He met some quite famous and interesting teachers, and the University of California at Berkeley. He is currently the Robert R. Bensley Distinguished Surface Professor of Anatomy at the University of Chicago, where it's very cold at the moment, I understand. and he's senior advisor to the provost to the University of the University on the Life Sciences. He blends the field of paleontology and molecular biology to really understand the history of life. He has led many expeditions to a large extent to many very cold places, and his team is widely known for the discovery of the Tiktaalik Rosia, the missing link, if you wish, between a fish and a life on land. And if for anyone who has read his latest book, you will know that it was this missing link. So how can life move from water onto land that really set him on the trajectory that he has been ever since? So this Tiktaalik, I don't even know if I pronounce it correctly, was a fish that sort of has, thank you, arms and legs, and therefore you can easily see how a water creature can start moving on land, and of course then the rest is history. So he is not only passionate about science, but he's also very passionate about communicating science, hence the wonderful books. But it also means that he has won several rewards. He has received a Guggenheim Fellowship Distinguished Service Award from the National Association of Biology Teachers, and the Science Communication Award from the National Academy of Sciences. And I couldn't agree more that we scientists have to do much better in communicating our science to a general public. Of course, you know that he's the author of 3 popular books. 2 have already been mentioned: Your Inner Fish, The Universe Within, and now then Some Assembly Required. He has hosted and served many senior scientific, he has hosted and served as senior scientific advisor on the Emmy Award-winning Your Inner Fish, a documentary, of course, based on the book. And he's currently a fellow of the American Association for the Advancement of Science, the California Academy of Sciences, the American Academy of Arts and Sciences, the American Philosophical Society, and he is an elected member of the National Academy of Sciences. So that's a whole mouthful. Before I hand over to Neil, and we can all listen to his wonderful talk, just to give you a little bit of information about the Q&A. Those of you who know Zoom know that there's a raise your hand option. We will not be using that option tonight. You can type in your questions whenever you want in the chat function, and then after Neil's talk, we will, depending on how many questions there are, we'll select some of the questions. The questions will be anonymous. I won't be calling out any names, so feel free to type in your questions whenever you want. And now it's my great pleasure to ask Neil to take the virtual platform and deliver his wonderful talk.
Well, thank you, Madeleine, and thank you, Daniel. It's a delight to be with you tonight. And I'm in Chicago in the afternoon, good evening in Berlin. Tonight, in about 45 minutes or so, what we're going to do is try to cover billions of years of time and to talk about molecules and DNA and fossils, and to tell us some of the surprising things they tell us about the history of life. I mean, it's a really wondrous story. You know, I almost take it for granted that as a paleontologist, I think in terms of millions, if not hundreds of millions of years. And yet it's hard for our minds to really grasp that vast extent of time and what happened on our planet and what happened to life on our planet in the extent of time that life's been here. Now, by some estimates, life has been on our planet, on Earth, for 4 billion years. That's an extreme estimate. Others have put it close to about 3.5 billion years. But either way, life got going very quickly after our planet was formed, because the planet was formed about 4.57 billion years ago. So life was around pretty quickly after Earth formed. And since that time, what we've seen are great transformations in the history of life. The first several billion years were transformations of single-celled organisms, microbes, experimenting with new ways of living, new ways of metabolizing, new places to live on this new planet. And then sometime around 700 million years ago, 600 million years ago, a great evolutionary event happened, whereas individual cells came together to make creatures with bodies, multicellular animals. And ever since then, the world has not been the same. you have animals, you have plants, you have fungi. And within each of those groups, you've had major transformations as well. Just take our branch of the tree of life. I mean, you go from worm-like animals to fish living in the sea for millions of years. Then at some point in time, fish evolved to walk on land, took those first steps into terrestrial realms. Then life began to take to the air, to fly, back to the water. All these great transformations in the history of life, which are so beautiful. And that's what I'm exploring. Let me share my screen, because what we try to do is we're living in a really remarkable age in science, where we have new discoveries from fossils, and we have new ways of studying DNA, in particular the DNA which builds bodies, which tell us about the great transitions in the history of life. And so how do we use these different tools to tell us about the way that life was assembled in the way it is? How did fish evolve to walk on land? Birds evolved to fly? How did our distant ancestors evolve to talk and to walk and to use tools? Just remarkable stories. Now, what I'm going to tell you is going to be actually somewhat surprising, because much of what many people think about evolution is actually wrong, including many people I talk to. And the motto here was when I was writing the book, at the end, I was thinking about sort of the great analogy to think about what's going on with these great transformations. Because if you think about, let's just take the transformation from like a reptile to a bird, it seems pretty impossible. How did critters evolve to fly? Or the transition from water to land? If you look at it, it seems downright crazy to think that way, right? But I was reading an autobiography of this woman, Lillian Hellman, as I was wrapping up the book. And this is Lillian Hellman. She had quite the life. She was blacklisted in the United States by the House Un-American Activities Committee for putatively being a communist. She had some very hard living ways. So when she was reviewing her own life, and writing her own autobiography, she had a quote that captures precisely what I'm going to tell you about tonight in terms of the history of life. In looking at her own life, she said, "Nothing, of course, ever begins when you think it does." That is a very, very powerful statement, not only about her own life, not only about the history of life on our planet, but also about the nature of history itself. Whether that history applies to political systems, social systems, or genes or development, what have you. Because the origins of structures, the origin of inventions, particularly in the history of life, never happens when you think it does. There are always antecedents, and those antecedents are not only surprising, they always come out before you would originally think they did. Now, I'm going to break that down. That's what the entire talk's going to be about. And we're going to break it down, and we're going to look at fossils, and we're going to look at genes. I'm going to show you that nothing ever begins when you think it does, and it couldn't happen any other way. Okay, so if you think that lungs evolved to help creatures walk on land, when fish evolved to walk on land, if you think feathers came about when the first animals began to fly, you'd be in very, very good company. You'd also be 100% wrong. And we've known this for over a century. Lungs did not evolve when creatures first took the first steps on land. Feathers did not evolve when creatures took those first flaps to fly. And this is true with every other great invention in the history of life. They always came about differently. Now let's break this down, because there's something not only a little bit obvious about it, but also something extremely unexpected. So here's a slide. This is the slide that launched my scientific career. I was a graduate student at Harvard, and I was looking for a PhD dissertation. And I was taking a course, from a leading light in the field, who was, every week was a seminar. They were talking about great transitions in the history of life. And each week would be a slide and a talk and a seminar on a great transition. And this was one of the weeks, I don't know, week 4 or week 5. And it shows in cartoon form what we knew at the time about the transition from fish to land living animal. What you see in the cartoon is a fish on top. It's that conical head and and a long tubular body, fins and so forth. And what you see on the bottom is a cartoon of one of the earliest creatures to walk on land. And both of these are cartoons of fossils that were discovered, in the last century or so. And what you see on top is a cartoon of a fossil fish from rocks about 390 million years old. We see on the bottom is a cartoon of one of the first animals to walk on land. It's a fossil, sort of amphibian, from rocks in Greenland that are about 365 million years old. I remember looking at this slide thinking, That is a first-class scientific problem. How did fish evolve to walk on land? I mean, because you think about what has to change to make this happen. You know, you have the transition from fins to limbs. Fish have fins, to locomote and move about in water. Critters living on land have limbs with fingers and toes and wrists and ankles and all that stuff. Fish, have lungs. Land living animals breathe air. Fish breathe in water. If you look at it, so many systems have to change that you'd almost think, well, all sorts of things have to happen simultaneously in the history of life for fish ever to take their first steps on land. And honestly, that's what attracted me to this problem. And indeed, I was thinking about it as a paleontologist. I wanted to find an intermediate. I wanted to find say a flat-headed fish with fins, with arm bones inside, that sort of thing. And we're going to talk about that in a second. But it's also just the intellectual beauty of the problem, right? Well, it turns out when you think about this, when you think about the inventions that creatures used to walk on land that enabled those fish to take their first steps on land, say around 375, 380 million years ago, there are some surprises here. And one of the first surprises begins with one of the great characters in all of paleontology, begins with Bashford Dean. Now, if there's one person who I'm very jealous of in the history of science, it's Bashford Dean. Dean had two of the greatest jobs simultaneously in New York City. You can see here is Dean. I think he died in the 1930s. I didn't put his lifespan there, but most of his work was done in the early part of the 1900s. Dean had a fascination with armor. As you can see, he's on the left wearing samurai armor, on the right wearing medieval armor. He used to walk around, he lived in Manhattan. He used to walk around the streets of Manhattan in samurai armor or in medieval armor, obviously to cause quite a stir. Or maybe in Manhattan that wouldn't cause a stir, but anywhere else it would cause a stir. But he was the curator of the medieval armor collection at the Metropolitan Museum of Art on Fifth Avenue of New York. Great, and he really sort of founded that collection, working with donors to build the collection. But at the same time, across Central Park, on Central Park West, he was curator of fossil fish at the American Museum of Natural History. That's right, simultaneously, he was curator of paleontology, fish paleontology, at the American Museum of Natural History on Central Park West, and of medieval armor at the Metropolitan Museum of Art on Fifth Avenue. So he'd shuttle across Central Park to do both. And interestingly, he was his fascination in fish was not just with any kind of fish. Guess what? It was for armored fish. He worked with armored fish in the fossil record. He was big into armor. Anyway, but he made some discoveries, and not only just discoveries, but he synthesized data that was around actually from German anatomists in the late 1800s, which have this long tradition of very descriptive and very detailed comparative anatomy, which is sort of the seed for much of the discoveries that people like Dean made. And that began with Gegenbauer in Jena in the mid 1800s and continued ever since. But Dean was sort of capitalized on and realized that there are a lot of fish like this. This is a lungfish. These are fish that are alive today. There are 3 different genera of lungfish. This is one from Australia. There are others from Africa and others from South America. Now, as the name implies, they have both lungs and gills. Now, what's amazing about these fish, though, is if you watch them, and this is the Australian lungfish, this is taken from one of Dean's monographs in the early 1900s. What they do is they use their gills to breathe to extract oxygen from the water most of the time. So they're pumping water over the gills and they're breathing with that. Every now and then, when their metabolic needs need it or the oxygen content of the water drops, these lungfish will go to the surface of the water and take gulps of air. They'll take gulps of it, force it in their mouth, and then force it into their lungs. And they'll do gas exchange that way. So these fish are sort of dual purpose breathers. They can use their gills in the water when the oxygen content of the water is sufficient and their metabolic needs are relatively low. But when they need it, they can go to the surface of the water and take big gulps of air and actually breathe with lungs. Now, Dean, as well as other comparative anatomists, obviously looked at the structure of these lungs, and they found that not only do they have lungs, But those lungs, if you look at them in detail, so you could see at the top, what you should see is you see a fish, and you can see in gray the extent of its lungs. It has a lung which runs actually a long length of the body. These are paired lungs. So what you see in A in this slide is a cross section to show that they're paired lungs, just like ours. And what you see in B are that the microstructure of these lungs is almost identical to ours as well. So these fish have lungs that are, for all intents and purposes, the same as our own lungs. And indeed, scientists working just published 3 weeks ago in the journal Nature showed that many of the genes, the DNA that builds our lungs, those are present in these fish actually building their lungs. So these are lungs, and these are lungs that are similar to ours. So what does it tell us? And Dean realized this very early on. What you see here is a sort of an evolutionary family tree. Okay, all the way on the right, you can see sort of an amphibian. Well, that's actually there to represent all land living animals. And then what you see on the branches as you sort of descend in this tree are the fish that are related to us, our cousins. So our closest cousins, as you could see, are lungfish. Then you go down, you see all kinds of other fish, including a shark shown in gray there, that are more distant cousins. Well, the reason why I'm showing you this is if you map the presence of lungs in fish, sort of on a family tree of life, here's what you see. You see that other fish have lungs. In fact, lungs are the primitive condition in fish. Lungs evolved originally in fish living in water, used along with their gills to breathe air when the oxygen content of the water wouldn't cut it. What does that mean? This is not just some esoteric fact of the history of life. What it meant was, when our distant fish ancestors took their first steps on land, they didn't need to invent lungs. Lungs were around for eons before, and their distant ancestors, ever before they took their first steps on land. Okay, so here's, Lillian Hellman, nothing ever begins when you think it does. Here, lungs predate the invasion of land by fish by eons, millions and millions and millions of years. And in fact, that holds for pretty much every other trait. Now I'm going to show you, but before fish ever took their first steps on land, they already had arms and legs. How do we know that? Well, let's go back here. Remember, I told you I was training to be a paleontologist. And so if you look, here you see that what we knew about the transition in the mid-1980s, from fish to land living animal. My team and I, looking at the right rocks, really set off to find one of the first creatures to walk on land, maybe a flat-headed fish with fins with arm bones inside, some mix of these creatures, of these features. What we did is we tried to identify places in the world that had rocks of the right age and type to hold these kinds of fossils. And we were fortunate in that we were able to find a few places. One of the best places to look, and this is what you're seeing is, for those who can't see it, a map of the Arctic Canadian province of Nunavut. So what you see on the upper left is Nunavut territory. There's a little flag in Nunavut up there with an Inukshuk. And circled in red is the northernmost province of Canada, Nunavut territory. And it extends all the way towards the North Pole. The main body of the slide shows the islands of Arctic Canada, from Ellesmere Island to Melville Island, beautiful islands. I worked there for about 13, 14 years. Circled in red here are where rocks of the right age and the rocks of the right type are present on the surface of the Canadian Arctic. We knew this, we found from the oil and gas literature from people who mapped these rocks. So essentially from 1999 to about 2014, my team working up in the Canadian Arctic really worked to try to find fossils that tell us about these transitions. These rocks up in the Canadian Arctic now were formed in tropical delta systems, tropical streams. So think about that for a second. What's in the Arctic now are rocks that were once formed in ancient tropical environments. The other place we work, and this is most recently, is Antarctica. And what you see here on the left is Antarctica, obviously the continent of Antarctica. And if you look carefully at that map, what you see is obviously most of Antarctica is the Antarctic ice sheet. But cutting across it, shown in brown on the map, are the Transantarctic Mountains. And there's a mountain range. It just literally cuts right across the continent. These mountain ranges actually poke through the ice. It's pretty wild. And we worked on them, following some geology work done by Australian colleagues and colleagues from New Zealand. We had a multidisciplinary crew, an international crew that included some scientists from Australia as well. we investigated a couple different areas here in Antarctica where those mountain ranges were poking through the ice, but those mountain ranges had rocks that were 380 million years old. Okay, so we basically would camp out on the ice, on the glacier, looking at those rocks, looking for fossils inside. And we would find, and here's a slide of Antarctica. This is one of the sites. What you see is the famous blue ice. You see that blue ice there? That's the old ice that actually holds meteorites when they fall. You can see the mountain ranges in the distance as they poke up through the ice. And what you see in the foreground here are green rocks. That's a green shale. If you look really carefully at one of those green shales, right in the foreground there, you see something white. That's a fish skeleton. So here, think about that for a second. In Antarctica, at the tops of the mountains, in the middle of Antarctica, in the Transantarctic Mountains, we're finding fossil fish living in ancient delta environments, 380 million years old. So what do they tell us? Well, and how do they fit into this whole Lillian Hellman theme? Remember I told you nothing ever begins when you think that it does, and that we were looking for a fish with a flat head and fins and arm bones inside, and working in the Canadian Arctic, that's exactly what we found. This is Tiktaalik Rosia. It's a creature about 1.5 meters long. What you're seeing here is only the front half of the creature. You'll notice it has a flat head with eyes on top. If you look at its back there, it has scales covering its back. And if you look in the foreground, what you can see are fins. See the fin webbing in the front there in the foreground. But this fish has a neck. No fish has a neck. You can see the head's separated from the shoulder. All kinds of other features. But inside the fin, when we cracked open the fin, what did we find? We found bones that corresponded to the upper arm, forearm, even parts of a wrist and digits, fingers inside this fish. Already a fish. And so just to show you this movie here, taken by one of my graduate students in a CT scanner just in Lumbar, what you see here is the fin on the right, and it's doing its little CT spin there. But what you see it as a humerus, one bone, followed by forearm bones, two bones, radius and ulna. And here you can see the blood vessels inside that map very nicely to blood vessels in our own arm. On the left, what you see is this is a fish that has shown in A, a shoulder. You can see the socket on the shoulder and the head on the humerus. In B is a fish with an elbow. There's the, you see the elbow joint for the radius and ulna. And then there are two different wrist joints, what are known in anatomical circles as proximal carpal and distal carpal joints. Now, what does this all mean? Well, if you look at what this creature done, this is basically a fish that can support its body using its appendages. It can do a form of a push-up, or it had a palm area of the fin, which can push against the ground. It had large pectoralis muscles, the chest muscles that can support the body. It had a wrist that can flex. It had an elbow that can flex, even and an upper arm that can support that body. This is a fish that had both fins as well as limbs with limb bones inside. It had a neck, it had scales. It's a mix of characters. So what do we have here? When you think about Tiktaalik, and you think about, and there's others, we now have 20 specimens of Tiktaalik, and Tiktaalik's not the only show in town. There are other fossils from southern Quebec, from Latvia and elsewhere that are very similar to Tiktaalik in many ways. But what you have living 380, 375 million years ago in ancient waters, in ancient streams, in ancient tidal areas, were creatures that had both lungs and gills, that had fins with arm bones inside, that had a neck. In some, that had pretty much all the inventions that fish needed to walk on land, to live on land. So basically the take-home message here to, harken back to the Lillian Hellman theme is that not only does nothing ever begin when you think it does, is that they always begins earlier. That our distant ancestors and fish, when they took their first steps on land, already had the major inventions needed to do it. They just were changing their function where they were used. Lungs evolved in fish living in water. Limbs evolved in fish living in water. Digits evolved in fish living in water. They're already good to go. And guess what? That story is true for almost every other great transition in the history of life. Just take birds. Look at this beautiful mandarin duck, gorgeous duck, right? When you think about birds in general, they have all kinds of unique features. They arose, 100 and 200 million years ago or so, but they have feathers, they have hollow bones, they have high metabolisms, they have wishbones, they have wings, they have all kinds of features that you'd say they're all unique. But guess what? Every one of those features is seen originally to appear in terrestrial dinosaurs. Feathers appeared first in dinosaurs. Wishbones appeared first in dinosaurs. High metabolisms in this lineage appeared first in dinosaurs, and on and on and on. It's just like the transition from fish to land-living animal, the transition from reptile to bird, same thing. All the inventions that the distant ancestor birds needed to fly already arose in a different context in dinosaurs. So the great inventions of the history of life all precede the revolutions that they're associated with. And really what happens is that to make these great transitions, creatures don't need to evolve new things. They already have them. They just have to change their function. This story applies to development as well. So if we think about this we all begin as a single cell. But when we think about where we are here in Chicago and in Berlin and elsewhere, we're all about 30 trillion 30 trillion cells in our bodies. So one of the great puzzles of biology, in fact, one of the great research areas of biology, is going from that single cell on the left to a multi-trillion cell adult on the right. We call that bodybuilding, where you have cells that are dividing, but also packaged in the right way. Turns out this story, to understand evolution, we need to understand how bodies are built and how that recipe that builds bodies can change. Right? So it's not just the anatomical structures, it's the recipes that build those anatomical structures in development. And this story begins, this is actually a long tradition in comparative anatomy, looking at embryos to understand evolution. It actually began in Germany with Karl Ernst von Baer and later on to Ernst Haeckel. But in France, there was a long tradition that began with Auguste Dumeril, who was working somewhere around the time after Darwin published his Origin of Species. Dumeril had something amazing happen to him. He was keeper of reptiles and amphibians at the Museum of Natural History and Zoological Garden in Paris. Okay, a great job. And he was, the recipient of all kinds of largesse. I mean, expeditions were going around the world collecting new creatures, and he would receive the amphibians and reptiles that they collected and study them anatomically, and now with Darwin's book, evolutionarily. So we got this shipment of 6 of these creatures you see here. This is one of the original diagrams. Oh, in the 1860s. And they were collected by a team working in Mexico. And they collected 6 of these because it was a funny creature to them. This is right after Darwin. And they looked at it and they said, well, here's like an amphibian, but it looks aquatic. See, it has gills, right? A frill of gills right at the base of its head on the right there. It has an aquatic tail. Look at that big flappy tail. It had all kinds of other features that suggested maybe this creature was a great intermediate between life in water and life on land. So that's why they collected 6 of them and gave them to Dumeril. Dumeril looked at them and said, that's pretty great. So he kept them, but he had other projects to do. So he put them in the menagerie, fed them, and an assistant would feed them. And salamanders, as many of you who keep them may know, they're really kind of low maintenance. You don't have to do much, other than feeding them and keep them at the right temperature and keep them humid. It's not that, they don't take a lot of care. Anyway, so the box was left alone for a long time. And they came back a few months later, and guess what? Well, they came and they were in for a big surprise. When they looked inside the box of the menagerie, they found not only these creatures, but a whole nother type of salamander. So here's a different one that was now in the box. How did it get there? No gills and did not have that aquatic tail. This is a very terrestrial salamander. Now it's like somebody put like a chimpanzee in a cage one year and came back the next year and found the chimpanzees living happily with gorillas in the cage as well. Did a new kind of creature appear out of thin air? Well, Dumeril was a very good scientist and he wanted to look at this in much more detail. And what he did is he looked at the embryology. And what he found is that it was a simple shift that happened in the box. So those aquatic adults there you see on the top, they produced offspring. There were males and females in there. They produced offspring, but the offspring that developed, they actually developed, but they underwent metamorphosis in their little enclosure in the cage in Paris. So basically, there's a simple shift in development. What you see is they would hatch from their egg, they'd grow as aquatic larvae. You can see those larvae swimming around like little tadpoles with an aquatic tail and those gills, and then the ones that Dumeril had did not undergo metamorphosis. They were basically became big sexually mature, larvae. But then the ones in the box, there was a little trigger, just because it was drier in the box, a simple little trigger in hormones produced this new kind of salamander. Same species, same genus, just a different version of it, which is remarkable. So what does this tell us? This tells us that a simple, tiny shift in the way that creatures are built in development, tiny shift in embryology, can bring about big changes in the adult. Okay, big changes. Look at the differences between these two versions of the salamander, but it's all a simple shift in a single hormone that can have changes across the whole body. Okay, so that's big, because now we can approach this from the level of DNA. That's something that Dumariel did not have in the 1860s. It's something we now routinely have when we think about it in modern molecular biology labs. And it really is a beautiful approach that dovetails very nicely with our thinking from paleontology. So it gets even better. Not only can we map the genes that are present in different creatures, we can begin to see how they function as bodies are built. And as this slide implies, we can use new genome editing techniques to manipulate those genes, to change those genes in certain ways, to ask some questions about evolution and other areas. Now I'm going to get in some details here because I think it's really worth it, because I'm going to get into an invertebrate, then I'm going to talk about us, and I'm going to show you how genes build appendages. So bear with me a little bit because I think it's worth it. A colleague of mine by the name of Nipam Patel has been working on this creature called Parhyali. It's a tiny little thing about 1 centimeter long, lives in sand, and it's clear, and it likes to jump around. Now, the reason why he likes it so much is if you look at the diagram in here, what you're looking at is a Parhyali, blown up. But look at it, has all kinds of different appendages in its body. In the front, it has, on the left, where the head is, it has antennae. As you go further back, you have larger claws. Go further back still, you have appendages that face backwards. Go further back still, you have appendages that face forwards. And go further back still, you have those little feathery appendages. So this is a creature that has lots of different kinds of appendages sticking out from his body. So Nipam's been studying the genes that build the bodies of these critters, okay? And he's been looking at the genes that are active when these appendages are made. And so let's just get into some detail here, but it's actually kind of simple. The idea here is that there's a map that you can make between the genes that are active in a part of the body of these creatures and what appendage forms there. So if you look here, we've color coded this. So look all the way on the left, you can see it's dark gray. Dark gray is a place where a gene called UBX is turned on. Where you have only UBX turned on, you get a backward facing limb shown in the dark gray there, okay? Now there's another region, as you go back further, you see the striped region where you have the front facing limbs where the blue arrow is. That's a place where both UBX and another gene, ABDA, are turned on. Okay, so there's a genetic address where only UBX is turned on, you get a backward facing limb. That's where the red arrow is. Where UBX and ABDA, this gene, are turned on together, that's where you get the blue arrow, you get a forward facing limb. Okay, and so there's a different map for different parts of the body, but that's the two places we're going to focus on, right? I hope you get that map, genetic map to what appendage forms there. Well, Nipam, using CRISPR-Cas genome editing technology, which won the Nobel Prize last year, Charpentier and Doudna, was able to manipulate this. And guess what he found? If you make a Parhyali lacking that gene ABDA, what happens? You basically go from having a creature like on top with both front-facing and backward-facing appendages to a creature on the bottom where you get rid of ABDA. We have turned the forward-facing segments that have forward-facing appendages into backward-facing ones. So basically, when he changed the genetic address in the development of these creatures using CRISPR-Cas genome editing, he was able to change which appendage formed there. Okay, that's pretty cool in itself, but it gets even better. Because guess what? We have versions of these genes too. These genes pattern the bodies of flies. Look on the bottom, you see an embryo of a fly and a body of a fly. These genes are turned on in flies doing similar things. Versions of these genes are present in our bodies, and they're active as our limbs are developing. They're active as our backbone, our vertebral column developing. And in fact, we can do the same experiment in mice that Nipam did in his creatures in the invertebrate I showed you in the previous slide. They've done it. So take the same genes, versions of the same genes in a mouse. Let's look at it. Here's a mouse. There's a cute little mouse. And what we're showing here is the vertebral column, the backbone, in the back end towards the tail. So zoom in on the bottom of this thing. What you see is a tail of a mouse. And there's two kinds of vertebrae we're going to ask you to look at: lumbar vertebrae and sacral vertebrae. And you'll notice there's a red arrow. That red arrow is pointing to where genes are turned on. Just like in that invertebrate, there's a genetic address for which kind of vertebra in our bodies develops where. And it's also seen in mice as well. So these genes in mice and humans are called HOX, H-O-X. And the two we're going to look at are HOX10 and HOX11. Where you have HOX10 turned on alone, you get what's called a lumbar vertebra. Where the red arrow is, you'll see where HOX10 and HOX11, another gene, are turned on, you get sacral vertebrae. If you manipulate this, just like Nepom did with his invertebrates, let's just say you make a mouse lacking HOX11, look at it, you can change a sacral vertebrae, a segment that has sacral vertebrae into lumbar vertebrae. So basically the take-home message of this complex example is that there's a genetic address that tells which appendages to form in different parts of the body of invertebrates. We share those genes too. And when we manipulate the genetic address of those, it can change in mammals, in mice, and in people. People haven't done the experiment, but we can look at birth anomalies, that changes in the activity of those genes are involved in changing the identity of different parts of our bodies, in this case, vertebrae. Pretty amazing. So really kind of nothing ever begins when you think it does. What's the take-home point here? The genetic toolkit that builds our bodies, that builds fish bodies, were already present in our distant ancestors. The same story with lungs, the same story with appendages, only now with genes, that the genetic toolkit to build our bodies is deeply ancient and has origins and roots in our distant animal ancestors. So I want to close with one sort of amazing example of nothing ever begins when you think it does. And that relates to the work of a neurobiologist, of all things, from the University of Utah. Jason Shepherd is a neurobiologist. He's from New Zealand, lives in the States now, but when he grew up in New Zealand, he grew up with a fascination reading Oliver Sacks's books. And he grew up with a passion really to understand the human brain, in particular, understand the development of the brain and how it works and so forth. So he, in graduate school, he started to work with a gene called ARC, A-R-C. And there's a reason why he dove right into that gene. This is a gene that in mice, when there's a mutation in this gene called ARC, they lose their memory. They're unable, they can solve a maze, okay, in one day, but they cannot remember their solution the next day. So it's a very specific deficit in memory. Humans that have a mutation in ARC, the gene, have a range of cognitive deficits, including a loss of memory. So he wanted to understand, what's the role that ARC is playing in memory. And he was, he's trained as a physiologist and molecular biologist. And what he did was he isolated the gene, and then he isolated the protein that the gene makes. Okay, that's what you do, right? You isolate the gene, isolate the protein, you begin to analyze it, understand its chemistry, what it does, and so forth. So he had the protein and he popped it under a microscope. And this is it. This is an electron micrograph, about 100 microns. And he saw, I'm going to show you this white arrow. He saw these little globs in the electron micrograph. See these little globs where that white arrow is pointing? Basically balls of protein. He noticed that ARC, the protein ARC that makes this memory gene, and this is from mice, makes these globs. And they have a very characteristic structure, these globs. And he was very curious. It rang a bell because he took a pre-medical curriculum in this and he remembered his microbiology. And he could swear he's seen these blobs that ARC makes before. So he's at a medical school at the University of Utah. So he calls some colleagues who are in the microbiology department who happen to work on HIV, the virus that causes AIDS. And he showed them the slide. And they said, well, that must be HIV, the virus that causes AIDS. He said, no, that's ARC, a memory gene. And here's what you're seeing on the right is HIV, the protein that HIV makes forms these globs that are identical to ARC in many ways. And what they did then is they started to decipher the genetic makeup of the gene ARC, the sequence, and compare it to HIV. They compared the structure of the protein that the gene makes to the structure of the protein that HIV makes. And it turns out that these are versions of the same thing. And then when they started to look at other animals, they found that other animals have ARC too, that seems to be related to HIV, but that if you start to look at fish, they don't have this as well. So the hypothesis came up that sometime in the distant past, a virus invaded the genome of our distant ancestors, a virus like HIV, but instead of causing over time an infection, what it did was domesticated and rerouted to make the protein that's now used in making memories. That's a remarkable story, that this gene that's involved in making memories in mice and humans and so forth actually has its roots as a virus. And that's not alone. the more we look, the more we see that not only some genes that are involved in brain function, but also involved in pregnancy and the formation of the placenta, they originally have roots as viruses that originally invaded. Indeed, when you look at our genome, when you think of DNA, fully about 9% of our genome is composed of ancient viruses that invaded our genome only to be defunct. Our genome is like 9 to 10% of it's like a graveyard for ancient viruses. Some of those ancient viruses were repurposed to do important new things. So they were, commandeered, if you will. But it turns out that if you look at our genome, only 2% of it is our own genes. The rest of it is all kinds of repetitive sequences, ancient viruses that are knocked out. When you think about how much genetic material is inside of us, it's just truly remarkable. I mean, think about this. We have, say, 30 trillion cells or so in our body, depending on the estimate. You can have different figures. But inside each cell is a 6-foot-long strand of DNA that's been packed and wound around itself to fit in the nucleus of a cell. If we were to take every piece of DNA in your body or my body, an individual of us, and unwrap it to its full 6-foot length and put it end to end, our DNA would run from here to almost to the planet Pluto. That is how much DNA is inside our body. It's just enormous computing power. It's just enormous amount of information there to build bodies, to make bodies work, and so forth. And this underlies evolution as well. What's amazing, as I said before, is if you look at that strand of DNA, only 2% of it are, categorically our own genes, the parts that encode for proteins. The rest of it are ancient viruses that have been knocked out, repetitive sequences that might jump around, and stuff that we don't even know yet what it may be doing. So it's a real mystery, but I can guarantee you that as we discover more about DNA and as we map it to evolution, what we're going to return to is Lillian Hellman to understand that nothing, of course, ever begins when you think it does.
Thank you so much. And I have loads of questions. So while other people type in their questions, I'm just going to be naughty and start with one. And I quickly tried to find the answer using Google, of course, but I couldn't. So Tiktaalik, I assume it was an egg layer because live bearing and fish evolved much later. Do you know?
Yeah, that's a good question. So we don't know much about its reproductive state. So basically, here's the cast of the head of Tiktaalik right there. So we have mostly the bones of it, right? But if we can compare it by analogy to lungfish or to other fish closely related to it in the tree of life, they're egg layers. And typically what they'll do is they'll have a clutch that varies in size. Like take a lungfish, they'll have a clutch that varies in size, from 5 fertilized eggs to 30, rarely more. And then they hatch as larvae. Some of them undergo metamorphosis, others don't. But it's an egg laying, not live birth kind of system. Although, there's a lot of variability we see in fish about these. So I'm hesitant to speculate too much about its reproductive biology, just simply because we have the bones.
But I guess because the larvae are always aquatic, they could not have reproduced on land. I guess the reptiles were the first to reproduce on land because they're of course a hard shell, isn't it?
That's right. So the famous hard-shelled amniotic egg is really sort of the final commitment to life on land. Now, creatures were already living on land before they were able to reproduce fully on land. So you had all kinds of creatures of that amphibious mode like Tiktaalik and some that are even more terrestrial. But remember, Tiktaalik and all these other creatures are still, and I think this is where your question's going, are still tethered to water for part of their life cycle, right? And so when we think about these things in sort of an ecological framework, we have to think about the whole life cycle, right? And so and because their life cycle varies, their ability to move about changes with size, what they may feed on may change as their head develops. Reproduction and larval life in water, it can be very different from adult life on land. So they may have been able to exploit different environments at different parts of their life cycle, and that's something, unfortunately, we cannot really sample too well with Tiktaalik Rosia. We have a growth series of them. We have 20 specimens, 20 individuals, I should say, lots of specimens, 20 individuals. The smallest one is probably about 1/3 of a meter long. The biggest one's about 10 meters long. Oh, not 10. The biggest one's about, I was about to say 10 feet long. So 3.5 meters long.
So that's pretty big for something that lives in water and on land. So were they mainly bottom dwellers, you think?
Yeah, I think so. I think they're benthic. Yeah, I think they're doing a lot of walking in the bottom of the water. I think that's kind of the main thing, but they can then live in the mud flats as well, using those same things that were useful in walking benthic underwater, would be useful living in the shallows where the depth of the water is probably equivalent to their body depth, and then on land as well, brief forays. But they certainly.
So the transition is not that huge, really. I mean, it's really a gradient.
Ecologically, you hit the nail on the head with that, because, and that's the big point, is that there isn't this huge dichotomy between water and land. That if you look at the transition from walking on the bottom, like living benthically, living in the sort of liminal area, right at the margins, and then to the true mud flats itself, it's sort of a continuum. It's not all or none.
Yeah. Anyway, I should stop asking questions and see what we have here. Well, one very practical question is, what's the title of the book in German?
The History of Life. The History of Life.
There are two questions which sort of related, and maybe we've already answered them in a way. So one person is asking, could you take the fish out of water and make it survive? But of course, we're not really talking about a fish anymore. We're talking about a transitional, unless the question is whether. Yes, you go.
Yeah, they are asking a very good question, because a colleague of mine did that. So there is a fish that alive today. It's closely related to lungfish. It's called Polypterus beercher, and it's this species lives in Africa, and it lives in that sort of liminal space. It lives in that space, benthic, shallow water. Doesn't really like mud flats, but it can, do that. So what he did was he forced them to develop in the mud flats, simulated in his laboratory. It's basically see what is the range of variation you have. If you raise them in a different environment, can you get something totally different? And he found that when he reared these things on a simulated mud flat environment, keeping them reasonably wet, so they could survive, he found that their shoulder developed in a slightly different way, and that it looked a lot more like a terrestrial vertebrate. So it's like they have this range of capacity in the embryo, and where they develop and how they use it can have big impact on their function. So yes, for some of these fish, you can rear them on land. And in this one case was a paper published in the journal Nature about 6 years ago or so. You can see that when you force creatures to do that, they during development can gain some, not all, some terrestrial features.
Yeah, because they must be able to get enough oxygen out of the air.
Through their skin. Well, Polypterus also has lungs. It's one of those fish with lungs as well. It can take gulps of air. Polypterus is great. It actually communicates on land. So it'll go in the mud flats sometimes and snort and huff and things like that. And there's presumably their mate recognition calls too. They use their lungs for that as well.
The next question is, what was the function of these appendages and lungs that evolved in fish before they transitioned to land?
Yeah, so that's a great point. So when you think about the environments that these creatures lived in back in the Devonian, it's a very different sort of world. So let me paint a picture of what the world looked like, some of these environments looked like in 375, 380 million years ago. So keep in mind, the rocks we're cracking are in Antarctica or the Arctic, right? So there are glaciers, there are polar bears in the Arctic, penguins in the Antarctic, different. But the world inside the rocks is this warm tropical world. What we know is it's very, very rich in fish life, these streams, these ancient streams. You have big fish, some of them around 10 feet long, like filling the crocodile sort of niche, big carnivorous critters. You had smaller fish that were armored. You had pretty intense vegetation. You had shrubs along the margins of these streams. You had some of the earliest forests, so you had tree-like structures. You had invertebrates living in both in the water as well as on land. So it's considered a very swampy, weed-choked, rich in life, lots of predators, lots of competitors in these environments. So when you think about what lungs and fins may have done here, they'd be very good. Remember, these fish aren't easily swimming around. We think these are fairly occluded environments, that they're fallen branches, that they're very complex environments. So the ability to walk in these swampy environments underwater or even on the mud flats probably was at a premium. So when we look at the structure of the fin, and this is the fin of Tiktaalik here. And so what you see is the humerus, radius ulnar, there's the digits, and there's the shoulder. If you look at the underside, huge crests. Yeah, big crest running out there. This is a creature that had massive muscles to support its body off the ground. So we're thinking that it was using these appendages to support itself, move about in sort of very complex environments where swimming in the open water wasn't easy to happen. Lungs for the same reason. When you have these environments, it's much like where we find the lungfish in Australia today, in Queensland. These lungfish tend to use their lungs seasonally more often than not. They'll use them throughout the year, but when there's rotting vegetation in the water, the oxygen content of the water drops, and they tend to rely on their lungs more during those seasons. So the lungs were used to breathe air when the oxygen content of the water wouldn't cut it. When it wouldn't cut it because the oxygen content is dropped for some environmental reason, or because maybe their metabolic needs are higher because they're doing stuff, and so it gives them that opportunity. So we think they're using both these structures for life in water, to specialize in their particular aquatic range of niches, rather than specifically for land. It turns out these features are really great, for when the time came to hit the land, but they already had them.
That's a great story. Someone doesn't quite understand the other part of your story, and that's the role of genetics. So could you, and this person is saying, back then there was no gene scissor. So I think you nicely illustrated the importance of particular genes for, the development of a body, for example. But how can you use genetics to understand some of the big steps in evolution? That's the question.
Okay. Yes. I see. Yeah, I see it right here. Yeah, it's a very good question. Thank you. It's Daniel. Yeah. So think about it this way, and I'll give you a different story because it'll explain it just the same. Many of the genes that build our hands are present in fish, making their fish fins. So just like where I told you the story of lungs, when creatures, took those first steps on land, they didn't need to evolve a new organ. Lungs already existed. The same thing is true at the level of genetics. The transition from fin to limb did not involve the origin of many new genes. It involved using old genes in new ways. That's the big piece, using the old to make the new. So yeah, the gene scissor didn't exist. Well, it did exist in bacteria hundreds of millions of years ago, but vertebrates weren't using it. No, but we're talking about, the major source of change in the genome, of animals and plants is mutation and recombination. These are ways that genes can change. And then natural selection makes some more common than others in populations. And that process can happen on genes that build bodies. So to explain it is, think about it this way. Many of the genes that are necessary to build a complex body like ours were already present in our distant ancestors. That's the take-home message. Many of the genes necessary to make fingers and toes were already present in fish making their fins. That's actually the same point as with lungs, right? Only we're talking about it at the genetic level. So I hope that explains it a little bit better.
Yeah, so the genetic architecture is just basically there. It's just then modified.
Yeah, we did an experiment. Exactly. We did an experiment in my lab a few years ago. I'm sorry to cut you off. I was just excited. We did an experiment in our lab a few years ago where we were able to take certain genes that were present in fish and put them in mice and see how they functioned. They functioned the same. When we put a mouse gene in a fish, it can function very similarly in the fin. So, some of these genes that are involved in fin and limb formation, we can take a human version of it and put it in a fish and see what it does. We won't put it in a human, but we can put the fish in a mouse and see what it does. And so when we swap these genes, what we can find is the fish version often recognizes the mammalian embryo. Likewise, the mammalian genes recognize the fish embryo when they're developing in it. So what that's showing is that, there's a deep history to these genes. And so it's minor changes to them brings about some of the changes that we see in evolution. You don't really need big changes to the genes.
Yep. There's a question about your phylogeny that you showed. So there's a big gap in the phylogeny between the first lungs and the lungfish. So what happened to the lungs in the intervening millions of years?
Oh, so basically, the most primitive sort of bony fish are presumed to have spaces in them where lungs were. But then if you look at cartilaginous fish in that phylogeny, they don't have lungs, they have gills. But then when you start to go to our branch of the tree of life, what you start to see, not only do lungfish have lungs, but things like polypterus have lungs, and other creatures have air organs in their next to their guts as well. See, there are two flavors of air organs in fish. One are lungs, the other are swim bladders, but they're both versions of the same thing. That's the important point. So this air sac, which a lot of fish have, which some of them use for like neutral buoyancy, it's called a swim bladder. That's a version of the same organ, which we call lungs, which are an air sac, which is used for respiration. So the idea here is you can actually show that the common ancestor of the fish that gave rise to us all had lungs. And if you take it further back to ray fin fish, they all had lungs as well. So I didn't show a very detailed phylogeny, but I could have filled it out even more, and you would have seen that, coelacanths like Latimeria has like a sac for lungs. Other creatures have a sac for lungs as well. And if they don't have a sac for lungs, they have an air bladder.
So it's either lungs or a swim bladder, right?
That's correct.
Because you need one of the other. Basically.
One or the other, but in our branch, it's all lungs.
Yeah, okay. There's a little bit of confusion about what Tiktaalik would have eaten, because some people are asking, isn't it supposed to be a herbivore because it was the first animal on land, but it's actually the first vertebrate on land, because prior to vertebrates moving onto land, there were a large number of arthropods and other creepy crawlers, probably much bigger than our normal creepy crawlers.
So if you look at, here's Tiktaalik, right? So you can see the teeth there. I don't know if you can see it easily. That's out of focus there. There are big teeth in this thing. So it has these giant fangs. If I was to open the skull up, which we did digitally with a CT scan, you can see these giant fangs. Tiktaalik was clearly a carnivore. And it had multiple rows of small teeth and then sort of one row of big teeth, about 5 of them, big fangs. We just published a study, about 2 weeks ago in journal PNAS Proceedings of the National Academy of Sciences to show how Tiktaalik fed. In fact, if you go online, you can probably see the movies we have of it. It was clearly a very effective predator. Whether it fed on fish or arthropods, probably both is the possibility. There were big arthropods, large invertebrates on land at the time, and likewise, in the water. So there were both smaller fish and smaller arthropods around that it could have fed on easily with its dentition. It also, interestingly, has a skull that's like a fish and that has cranial kinesis, that is, bones in the head can move relative to one another. So in water, it could take big gulps and bring in prey. On land, it could snap the prey like an alligator. So it's sort of dual purpose.
Sounds like a wonderful creature.
Yeah, you wouldn't want to swim with it. No.
Probably not.
No.
Someone is asking if you could find another missing link in a fossil record, which would it be? Which of course is a bit difficult because it's a bit unclear what the missing link refers to.
Oh, I could tell you what I'd really like to find for, and it's always been on the burner of something I'd really like to do paleontologically, would be to find the earliest vertebrates, creatures with backbones, skulls like ours. I mean, there's some good fossils out there from China. There are a couple good fossils from northern Greenland, but we could do a whole lot better. There's a big gap in our knowledge there. It'd be nice to have early vertebrates. Some of the first for the creatures with, our kinds of skeleton, our kinds of backbone, that kind of thing. I'd love to do that, and if to do it, you'd probably want to focus on pretty, rocks much older, obviously, than we do with Tiktaalik. And there you'd probably want to focus more on marine rocks, ocean, near shore marine rocks.
So it'll be busy for a while.
Yeah, probably.
Sorry, I'm also trying to read all the questions. Someone's asking a question, what do we know about the dynamics of evolution beyond what Darwin describes? That's probably another hour's talk if you want to address.
Yeah, but I can touch on that because there's some really cool stuff that, look, Darwin was working, what's amazing about Darwin is he was working in an era before genetics. Didn't have genetics, right? We all know that. Mendel was years later. Population genetics was years later. DNA was, a century later. So all his work came about in an era where we didn't know much about how information was passed from generation to generation. He knew of animal and plant breeders. That was important, evidence in origin of species, but he didn't know the dynamics of the genome. Or even the concept of the gene, it was years off. So when we think about some of the surprises, I think that we would have in the over century since Darwin, century and a half since Darwin, some of the biggest ones that happen are kind of like the viral story, which is that information can be passed from critter to critter, right? I mean, we're used to thinking about passing information from parent to offspring in evolutionary terms, right? But the more we look, the more we learn. In some settings, information can be shared. In microbes, we know that, but also in vertebrates, information can be shared through the viruses and other modes. So, I mean, I think that part of the story about how information can be shared, genetic information, molecular information can be shared from lineage to lineage is really big. The other big thing, and this is, I think, I don't know where it's going to end up yet, but it's still it's very exciting, is that we have in our bodies, in our genome, a number of genes that just like to make copies of themselves. They make copies of themselves and they jump around the genome. And what they do is they make copies of themselves and they'll jump in another part of the genome. And depending on where they land, they may disrupt the genome, or sometimes they may be responsible for a new trait. So these jumping genes are playing a very big, increasingly our knowledge is showing they're playing a very big role in evolution. The question is what and how. It's thought that there was a paper just published like this week, actually, that maybe the origin of many new genes may be due to how these jumping genes work. But anyway, the thing to think about is Darwin had no idea of genetics. And then the notion of genetics we're getting in the last several decades is some real surprises. That the genome, our genome is roiling all the time. Viruses are invading it. Different parts of it are jumping around and landing in different places. It's a real riot of activity in the genome, and the ways that translate to evolutionary change, I think we're just beginning to decipher, honestly. And so I think when we think about evolution post Darwin, I think it's the molecular and the genetic changes. I think that would have surprised Darwin the most, honestly.
So these jumping genes, so you say they copy themselves and they also jump. Well, that is different again from just normal gene duplication, isn't it?
Yeah, exactly.
Yeah,
so
okay.
Normal gene duplication is, like polyploidy in plants, where something goes wrong when cells are dividing and you can end up with extra chromosomes. There are other ways that happens too. You can have whole segments of the genome that are duplicated. This is different. These are elements, transposons, transposable elements. They just make copies of them. There's a couple flavors of them, but in general, what they do is they jump, make copies of themselves and land somewhere else. There's differences, but they're very dynamic, and so if you look at our own genome, about 2/3 of our genome is made of these jumping genes that have just duplicated and copied themselves and land. So it's like they're almost like taking over the genome, in fact, it might even be 2/3, it might be more than that. I was basically relying on an old estimate, honestly.
And they do not have a viral origin. I mean, the real human, okay.
No, they're not retroviruses.
Yeah, okay. So these retroviruses are, and there's a few questions about retroviruses too. So you think most of the retroviruses are successfully suppressed, or do you think that many of them actually have a function like the ACE one that you showed? Yeah. But we just don't know the function yet.
Yeah, good question. I don't know. All I know is that almost 10% of our genome are dead viruses. So that means over history, they've just been, again and again and again, this path of them. I do have a strong suspicion that the incidence of sort of viral origin for important features is only going to increase, because the technology to find it has gotten, it's computational largely. You're comparing sequences computationally among different things, and we now have those tools, and the databases are all out there. So I think that's sort of inevitable. We're going to see a whole lot more of that. To get directly to your question, you know, what fraction of them are likely to do this, I honestly don't know. But I do know that viruses, yeah, they're, you know, you know, if you look at the number of viruses on our planet, and that includes not just in bodies, but in the oceans and elsewhere, there's more individual viruses on our planet than there are stars in the known universe. You know, that's like, that's that Carl Zimmer, great journalist, came up with that analogy a few years ago. That's sort of that, and it's very powerful. So when you think about just the sheer number of viruses and how much they mutate and how much they can impact our lives, it wouldn't surprise me if their, you know, if their extent is even greater than we can envision right now, honestly. So I think we are seeing a bit of a tip of the iceberg with viral impact on our history and our bodies. Now it's a useful question. Yes, people think about, you know, well, SARS-CoV-2, the virus that causes COVID-19, you know, can that be useful somehow, right? Unfortunately, that's a different kind of virus. That's a different kind of virus that doesn't enter the genome. What it does is it enters the cell and it commandeers the genome to do its dirty work, right? But it's not entering the genome itself like HIV and some of these other viruses.
But so CRISPR-Cas9 basically came about from people realizing that bacterial genomes have these repeated sequences, which are basically viral sequences, and isn't it? Or am I confusing myself?
No, you got it right. I mean, it's basically a bacterial immune system.
Exactly. So, and the fact that even our genome, and probably every single living organism's genome, is also full of retroviruses, could that also have some sort of effect on or immune function? It's just basically the theory of the genome, and if the organism experiences that particular virus, then it can mount an immune response. Which is wondering if that's one of the reasons why these viruses have been incorporated.
Yeah, I mean, I don't know. I mean, so you know, we have an immune system that's different from the bacterial one. The bacterial immune system is a version of it is this CRISPR-Cas, but there are other ones out there that, you know, they recognize a pathogen, they clip it up, it's done, and then they keep a memory of it somewhere around, right? So, but no, there's definitely a feedback, you know. So I mean, the question about Darwin, you know, what would Darwin be surprised about, or what's, you know, what do we need to modify post Darwin? I think it's kind of that kind of thing, that we're used to thinking about the genome as being this sort of just this lockbox of information that only changes via mutation and recombination, as we teach our students in introductory evolution. But there are really all kinds of environmental effects on the genome that can be carried on for, you know, for generations. One of them is viral invasions. Some epigenetic changes, some environmental changes can cause slight molecular changes to DNA, and in some cases even RNA, that can go to different generations. You know, I mean, are those significant and huge new mechanisms of evolution? That remains to be demonstrated, but they're definitely surprises relative to where we were in biology, you know, a couple decades ago.
But it does seem a bit weird that our genomes are so full of these viruses. I can imagine from the viral genome point of view, it's a great way, because you insert yourself into someone else's genome, and if you go, you don't have to do anything anymore. But you know, we were always taught that having a genome is expensive, because you have to maintain it, you have to duplicate it every time the cell divides, etc. So, do you think there are any benefits to having all these so-called junk DNA, if it really is junk DNA, or do you think it's likely, as we discussed earlier, that it does have a function, we just don't understand the function, or genomes are actually much cheaper than people claim them to be?
Yeah, good question. So, I mean, and obviously it's a hot button debate in the field. I tend to fall along the functionalist end, and, you know, people disagree, and it gets really kind of hot. But I think, you know, nature is, you know, if a trait is not useful, even if it's neutral and has a cost, it's going to disappear over time. So as you're saying, the genomes are expensive, because, you know, a cell has to go through the cell cycle. It's got to do a lot of stuff to duplicate DNA, particularly in a lot of genomes, particularly in the cases where the genome's huge, like the salamanders I showed you. They have giant genomes. Giant, and it's loaded with these repetitive sequences. So my sense is they're going to be doing something, otherwise they would have disappeared a long time ago, or there may be a cost to getting rid of them. You know, you
So,
keep
or
in
you
mind,
can't get rid.
yeah, or if you can, keep in mind that the you know, there's more to DNA than just the genes and the elements that control the activity of genes. It's the higher order structure of DNA. So one thing we're seeing now is, remember I told you there's a 6-foot long strand of DNA inside every, you know, rolled up on itself inside the nucleus of all our cells, right? But the thing about it is that that ball is very dynamic. And so it opens, it closes, it twists around, different parts of it touch each other. So the structure of DNA inside the cell at a microscopic level is amazingly dynamic. And the reason for that is genes are continually, you know, during this talk or during our lives, are continually being turned on and off in different parts of our body, particularly during development. Genes are turning on and off during development to make organs and things like that. But part of the turning on and off of genes involves these higher order changes in the configuration of the DNA molecule within the nucleus. So it's doing these complicated dances inside the nucleus and changing its shape. And that structure and that shape matters a lot, you know, to how the DNA works. So my hunch, and I'm just guessing, is some of this stuff may relate not necessarily to being a functional gene, but being part of the dynamic 3-dimensional structure of the genome itself. Maybe they get incorporated into how the genome actually does this 3-dimensional dance as genes are turned on and off. Maybe that's why we can't get rid of these extra genes or these large swaths of, you know, repetitive sequences. That's just a huge guess, mind you, but I'm just saying that there's a lot of unknown about the higher order structure of DNA, and perhaps some of this stuff, this junk may fit into it, may make more sense at that higher order level than the, you know, the level of the sequence itself.
What struck me in your talk is that most of the people you talked about, including yourself, have sort of worked across different disciplines. I mean, you know, the last part of your talk now, you're basically talking as a molecular biologist, whereas I know you started as a paleontologist. So I find that interesting. And I think for the new generation, making those big links and jumps is probably much more difficult than it ought to be. Because,
I agree with you.
Fields are becoming so specialized, it's impossible to even keep up in your own particular field, let alone make those connections between a different field. And then if you're interested in something like evolution, you have to understand at least the basics of all those different subfields. And I think we're doing ourselves a little bit of a disservice by overly specializing. Would you agree?
Oh, totally. I mean, so I look at my own life history that way. I trained to be a paleontologist, as I said, you know, so I was, you know, working with Stephen Jay Gould and other people, and I was working with field paleontologists and geologists and all that. And then, like in the late 1980s, just as I was beginning to launch my own independent career, all these papers from molecular biology started coming out, showing, you know, the genes that are involved in development and how they're similar between people and mice and fish and flies. And I remember thinking, I got to change your diet. So I'm thinking, it's like, I'm going to go extinct if I don't change. So, I mean, for the questions I wanted to answer, I mean, paleontology is great for certain questions, but for what I wanted, I needed both paleontology and the genetics. So I was fortunate to be able to take a sabbatical and learn techniques, and, you know, and try to retool a little bit to bring that into my own field. But as I'm sure you know from your own work, everything is now so interdisciplinary that you have huge collaborative teams now involved in papers with maybe computational expertise, informatic expertise, anatomical expertise, ecological expertise, what have you. And science is increasingly, you know, the important questions of science are, and many of them are increasingly interdisciplinary. And I don't think we do our students a giant favor by this super specialization, you know, right into a thesis immediately without any sort of training and exposure to other fields. And I think in evolution, we might have a little advantage there, because evolution, you got to learn behavior, you got to learn anatomy and embryology and all these different components of it. So evolution itself is a very, you know, it takes you to all branches of biology, honestly, because it impacts all branches of biology. But thinking about how to train students in this age is actually challenging, because you need to set them up not only for doing multidisciplinary work in their careers, but also, they're during their graduate careers, but also giving them the tools for lifelong learning, so that they're a little fearless about entering other fields or developing collaborations. So have something that they can carry with them, throughout, because it's only going to get more in the coming decades. So giving them a sense of fearlessness, giving them a sense of the notion that how to start collaborations and what how to go about that, because it's hard for any individual to, as you said, keep pace with their own field, let alone all these other ones that are out there. I always feel like I'm drowning, in many ways. But again, it means building collaborative teams and learning how to manage that. And, sometimes collaborative teams aren't always easy to manage, and learning how to manage that part of it. But I think we needed to school ourselves in, interdisciplinary collaboration in many ways, and that has a social component as much as a scientific one.
Let's stick to the science, because some questions came in and we have a few minutes left. Someone is asking or mentions that the pufferfish has a very small genome. So how did it get rid of the extra sequences?
Yeah, that's great. So that's a good, I don't know how they got rid of them. They have a super small genome. And in fact, they were originally proposed as a great genetic model by the late Sidney Brenner, Nobel laureate, because of their small genome. Turns out they're missing a lot of key genes, and but they've swapped functions among them. So if you look at fish, here's what's interesting, is most major groups of modern fish have a whole genome duplication. They basically, where we have one gene, they have two. However, they've lost genes in the process. Now, when we talk about losing genes, that can happen in several ways. You can actually, when a genome is duplicated during what we call meiosis, when they're making new cells, you can actually have a mistake which deletes a whole portion of a genome. But there are other parts where a gene can be deleted simply by a mutation that makes that gene non-functional. So the gene will still be around, but it'll be non-functional, kind of like with viruses. But with fugu, with the pufferfish, known as fugu, what you clearly have is something that's probably happened in meiosis, where they've actually deleted whole chunks of the genomes at different times. But there's another big piece of that, is they've swapped functions among genes. So I think that would be, if somebody wants a great PhD thesis, that would be really, really cool to see how gene function has evolved as something like fugu and the pufferfish and others have lost genes.
So they're more efficient, basically, with their genome.
Yeah, they're incredible. They make fins. They make very good fins, but they're missing some of the key genes that do that. So,
well, the last question is sort of related. It goes back to humans. So the question is our genome still growing because of these jumping genes, or has it been stable?
No, there's nothing stable about our genome. Yeah, it's you come back in 10,000 years. We may look kind of similar, but our genomes are going to be pretty different. There's stuff hopping around and moving about. There's a war going in genome between these elements that are selfish, that just like to copy themselves, and then other parts of the genome that want to restrain that. And with viruses invading and all kinds, so we think about what we are. We're essentially 30 trillion cells of our own cells, but we got an order of magnitude more cells in and around us of microbes, whether it's viruses or bacteria. So we're mostly bacteria and viruses is what we are. And our genome is living in a balance with those bacteria and viruses that are inside us and outside us. And changes to our genome are happening in that dynamic balance as well. And we change that, with our own practices, but, nature changes that as well. So yeah, I think our genome is continually growing. There's definitely changes still happening in our genome.
So next time you look in the mirror, and this is for everyone, and just realize you're not actually looking at yourself, or you have to ask yourself, what am I? But that again is a different topic. It is almost time. I think the discussion has been wonderful, could go on for hours, but I've been told that I'm not allowed. And I would thank everyone who has asked a question and who participated, listened. I hope you all learned as much as I did. And do read the book, either in German, which I haven't done because I couldn't, and also I wouldn't because the English version is much easier. It is a wonderful book, and I hope you will write many more. Thank you very much.
Thank you.
Well, thank you both. Thank you, Neil Shubin and Madeleine Beekman, for an incredible talk. I mean, I've been on the edge of my chair and really enjoyed it all. I know that I am going to be lying awake all night thinking about jumping genes and that plenitude of viruses. And I will get back to you if I can't get back to sleep this week. Let me just say, I hope that everyone will now, if they don't already have a copy in English, will run out and buy the German edition of this book. You can order a copy via the website of S. Fischer Verlag. There are probably even other websites where you can buy it too, but that's where we're telling you to go. So we hope that you will get it, and it is available today. And Neil, we hope that you will come to Berlin for an update talk on when you get around to the 10th edition, which I'm guessing is not going to be very long because it just sounds completely fascinating. Before closing, I just want to add that our next event will be tomorrow night at 7:30 Central European Time, 1:30 Eastern Time, and consult your global clocks for if you're elsewhere. Our Holtzbrink Fellow, Nagme Sarabi, will be giving a talk entitled When Love Was Forbidden, Sex and Intimacy in Iran's Revolutionary Generation. And please do register on the Academy website. We look forward to welcoming you. Maybe we can get Neil to address, the jumping genes in that context as well. Anyway, this has been fabulous, and I'm only sorry we couldn't do it live, but this has really been incredibly interesting. Thank you, Neil and Madeleine, and thank you to the audience for joining us.